Biophilia is a real thing. There's a reason why it feels really good to go for a walk in the forest. And I think part of that feeling good comes from our body registering the fact that we are being exposed to these diverse microbial populations.
Hey, Karl, how's it going?
Usually I ask you and you're like, there's a lot going on. We've been going to a lot of events. And then I say something like, yeah, but we're also grinding away at work. But this time I'm going to start by talking about going to a bunch of events. And really the one event that I want to call out was the founder-led Biotech tour that was hosted by Tony Kalisa and Pillar Ventures. So Tony of Pillar has been on the podcast in the past. It was my first time meeting Tony in person. He and I have known each other for a while, but it was really a good event. I thought there was a lot of energy. It was great to see a lot of old friends and new ones and make new ones. I thought the crowd was a little on the young side, but I'd love to get your take on it.
Yeah, I went to the event last year and yes, it's an incredible gathering of people. Some of them are early stage startups. So I think that's where the younger side comes from. But there were startups by people that were a little bit more seasoned that I actually brought to the event. And it was great. Tony gave a speech about how everyone in the room was very passionate about biology and that we all need to work together and move biology forward. And it was a great speech. I think people were shouting and whooping and they're like, this is awesome. Having all those like-minded people in that space. It was at this like rooftop bar. It was packed. I felt like I was at a club or some concert. That's the vibe. That's the type of people that Pillar bring together. They bring a great, strong community of people, founders, investors, supporters. We're part of that community. I can't wait for the next one. But this founder-led biotech tour, it has 4 or 5 stops. So this was New York, but they have SF. Last year they did London. So they're going to different cities. We'll put a link in the show notes. So I think they have like maybe one more event left, but it's good to keep track of for next year. And if you don't follow Pillar, you should follow them immediately. If you're an entrepreneur looking into biotech across biopharma, across bioindustrials, they're again very founder focused and they understand that it's the founder that makes the company, not just the technology, not just the industry, but founders who are motivated, that have a clear vision, that are coachable. I think that's a word I want to triple underscore. So they really foster this community and I invite anyone to join. I hope Tony's okay with that. I'm inviting our listeners to join the Pillar community. But yeah, I love it.
So one of the things that Tony said that people say, and I think it's worth repeating, is that companies that are started during tough times, we're not in a tough time, though people are predicting that tough times are ahead. And we have been waiting for an economic downturn, hopefully a soft one, for a long time. But we did recover from a pandemic that basically brought the economy to its knees. But Tony reminded the people in the crowd that companies that are started during tough times are the companies that tend to succeed because it's much harder to raise money, it's harder to hire people, and it's harder to be successful. So if you are able to succeed during those challenging times, you're more likely to be successful. We've seen people come and go. We've seen the community shrink and grow and shrink. We've been able to make it through those tougher times, and that requires a certain amount of resilience. So if you're thinking about starting a company now, go for it. It's always a good time to start a company. Then there's other events this week, Erum. I think you're out more than I am. I'm grinding away, not that you're not, but let's talk about the events that are going on this week.
Yeah, well, a lot of the events I'm going to more recently is to support the Merck Digital Sciences Studio. So at Messaging Lab, I'm the advisor for the Merck Digital Sciences Studio, and they have an incredible cohort of companies this year. I will also put a link to them in the show notes because you'll be able to see how these companies are innovating across drug discovery and development to really help accelerate therapeutics getting to patients, right? It's a very incredible group and program as well. And so they were at the founder-led biotech event last week, which was a lot of fun. But this week they have a couple events that they'll be co-hosting. One that's happening, actually, they have one event. They have one event that's happening this week, which they're doing in conjunction with LifeX VC, which is a fund focused on longevity. they're having this AI and bio event, which this term is being used a lot in the events that at least I'm attending. There's another AI and bio event happening this week, which is led by some startup founders. We went to one of their events. A friend of mine who's a machine learning expert worked in some of the big e-commerce companies, but she's been always very interested in biology. So we've been talking. She's now a mother of two, so we have that in common. I mean, just incredible, smart human being. And she's like, let's go to this AI and bio event. And I was like, sign me up. So moms are going out. We're going to go and network on AI and bio. So don't wait up. But besides that, there's a lot of activity. There's been a lot of tech activity in the space. I think people are starting to now, again, post-COVID, really embracing the in-person events, because that's when you really get to spend quality time with someone else, look into their eyes, get their vibe, see if there's a fit that can then lead into a stronger relationship. And I think that's why people are gravitating towards these events or hosting them, because there is a clear ROI, whether it's more awareness for what you're doing, or you're finding a potential partner or team member or customers or investors. It is amazing to see that there's this resurgence of events, and particularly in the space of AI and bio, which is super complex.
So tell me again who you're looking to meet at this event, Erum.
I'm always looking to meet people that are founders or that are working in industry, that are just looking to be more innovative, looking to see if our friends show up, to see how active they are in the industry or learning about these topics.
It's great to hear what you're looking for. I'm going to go see if I can go find those people at the event now, and I'll bring them back if I find them.
Are you trying to end this conversation with me? That's Karl's tactic.
So anyway, speaking of AI in biotech, this is something that we're living a lot. And there was two things that we wanted to talk about when it comes to AI in drug discovery. One is that NVIDIA just released publicly their BioNeMo framework, which is an open source framework for helping scientists with drug discovery. We'll leave that in the show notes. I think it's very exciting. If you're interested in learning more about NVIDIA and their efforts in biotech, definitely listen to the episode with Vega Shaw of NVIDIA. It's actually one of those episodes that performs super well. A lot of people have listened to it, and Vega is so well spoken.
All the shareholders probably listen to it.
Right. We're also seeing the applications of AI in consumer biotech, which is something we talk a lot about here. But one of the things that you mentioned as we were going to this conversation, Erum, was about prompting. Prompting ain't easy is what you wrote. And I'm just going to mention, I have been asked a number of times, hey, are you worried that AI is going to take over your job? Meaning, because a lot of the work we do is strategic thinking, a lot of writing, we spend a lot of time with ideas. And I'm always like, there's no way, because what we do is so specific, and the companies we work for are always on the bleeding edge. And you got to know how to prompt to get anything out of these large language models. Does it make us faster? I think it does make us faster. Does it bring out things that we maybe haven't seen? I think it does. I know that you call me out sometimes when you're like, don't use AI here, or don't deliver this because it sounds too much like AI, which usually doesn't happen because I am a writer. And so I'm pretty good at masking AI. Now I know there's tools for checking whether it's actually been created by AI. So I use that as part of my process. But you had something very specific you wanted to mention when it comes to prompting ain't easy.
Yeah, prompting ain't easy. I will say depends on who you are and what you're doing. I equate prompting with creating a hypothesis, asking questions, which people think, oh, finding answers are hard, but setting up a hypothesis and asking the right questions is equally difficult, right? If you're not asking the right question or the right prompt or have the right hypothesis, you're going to go down a trajectory where you're not going to find the answer that maybe you feel is what you were looking for. You might in your head have a clear understanding, but then when it comes to taking the idea that's in your brain and framing it in a way that can be understood by whatever AI tools you're using, it might not be as accurate to what you're thinking. I'm specifically thinking about our audience or people that are starting to understand that biology is super valuable to their industry.
Right.
And they want to be able to leverage AI to be able to maybe formulate a product or understand how the work that they're doing can be enhanced through biology. And I've seen people trying to do this and they're like, oh, it wasn't really helpful. But then I'm like, well, what question did you ask?
Right.
And there has to be some level of understanding how biology works, which I know no one knows how biology works. But obviously, you have high school understanding that there's the central dogma of biology, how DNA codes for proteins and how proteins are differentiated, things like that, where you are able to ask questions on a more biological level. And also depends on, obviously, which AI tools you use and what data that they're analyzing or access to. Of the different flavors of AI platforms, from the chat GPTs to the Perplexity to Claude, and I know Perplexity is getting mainstream time. Like, people are saying that has been shown to be better because of the way it cites sources. And I'm sure it has access to some great databases that have research papers in them. But I think that being able to ask the right question in your inquiry is going to unlock huge insights. If you ask a question and you get something, you're like, okay, that's what I understood, or like, that's not really telling me a lot. It's not that AI per se, it's probably you not being able to formulate a question that's correctly. Because I've seen this. So let me just not blame our listener, but blame myself, because I've been toying around and I'm like, I knew that, or like, that's not really telling me much. But then I went through this inquiry about the skin microbiome, and I was really asking about like cellular interaction and like the different layers of cells and what type of delivery mechanisms can penetrate the dead skin cells. Then I started asking like, okay, well, what products in the market have this type of formulation? What are the costs associated with developing that product? Because I was just interested. We had the conversation with Rocky Grazios of Estee Lauder a few episodes ago. So then I've been thinking about skin a lot. And so then I was just like, all right, let me just have this conversation. It's a very freeing exercise. It's very delightful when you start then learning with AI. I'm like, okay, so tell me a little bit more about these cellular interactions. And I'm like, oh, okay, I didn't know that. Oh, all right. Well, if I were ever to go down this route of creating a skincare product, I would at least know where to start or continue that line of inquiry. So that's all I really wanted to say about AI in general.
There's a lot to say about it, and we can have a whole episode on that. You mentioned Perplexity. I tend to use, it's because of you, I use Perplexity as a research tool. What I find interesting about using it as a research tool is sometimes the results are things that either we've written or our clients have written. That has happened.
Oh, really?
Yeah, that has happened more than once, which I find fascinating, which also to me says that the content that we're creating is often on the leading edge. And so Perplexity doesn't have references to that content. And a lot of the content that we create, we do use scientific papers to back us up. The other thing about Perplexity that's interesting is if you're using the Pro model, Pro actually now will shop for you. So you can do shopping within Perplexity. And I know that you had on your list of things to go to is like a use of AI agents, which is something that I'm super interested in and am looking to learn more about. I feel like we have barely even scratched the surface of this stuff. And yet I find that it's really interesting that it's become part of my workflow as a writer. I use it almost every day. Actually, I've replaced a lot of my political media consumption with AI and learning how to use it better.
That's good. That's better use of time. So I was telling my husband, oh yeah, I'm going to this AI event and this AI event, blah, blah, blah. And we were drinking coffee this morning. He was like, how does a fish know that it's in water? Or something along those lines. It got really deep. And I think what he was alluding to is we're getting deep into this, but let's take a high level look at what does this all mean? Where is this going? Right. And that's a question for our audience to ponder. We'll leave that in your brain. But someone who is familiar with AI and computation is our guest today, Elizabeth Henaff. I'm so happy to introduce her because she is one of those leading thinkers of biology and computation. Anytime I speak with her, it's just like, you did what? You learned what? So Elizabeth, she's a computational biologist with an art practice. So how cool is that?
Yeah.
And she has a bachelor's in computer science, a master's in plant biology, which we need to hook her up with Rocky Grazios of Estee Lauder. They can both nerd out on plant biology.
Totally.
Yeah. And she also has a PhD in bioinformatics from the University of Barcelona.
Wow.
It's really cool. But Today, she holds an assistant professor position in the Technology, Culture, and Society Department at the NYU Tandon School of Engineering in New York City. So she is in Brooklyn, technically our neighborhood, but just a few stops away. She's a friend of our little Brooklyn biotech mafia that we have here. I personally got a chance to talk to her regularly at different events and had lunch with her. I think this is the first time you've met her, right, Karl, on this
I think
episode?
she and I have met at different events, but I think you know her better than I do.
Yeah,
for sure.
But now you know her, and we're going to hang out with her more. Let's start our interview with Elizabeth Henaff.
Let's go.
Elizabeth Henaff, we're so excited to have you on the Grab Everything podcast. Welcome.
Glad to be here.
Yes. Well, I've been wanting to have you on the podcast because we have been running into each other at a variety of events, from the Biodesign Challenge to SynBio Beta and other events that have been happening around Brooklyn. And luckily, finally had a chance to have lunch. A veteran of the podcast, Ginger Dozier, she's a 2-time veteran, so she was on the podcast twice. She joined us and we had such amazing conversations. And I was like, I wish I had my recorder to capture what Elizabeth is saying, because you talk about some of those fascinating work that you've done with your students at NYU. So thanks again for being here. I really appreciate it. So let's kick off the conversation, because what we've been really interested is in environmental microbiomes. And your work centers around environmental microbiomes, especially in the urban and built environments. To kick it off, what sparked your interest in exploring microbial life within human design spaces?
The first time that I heard about environmental microbiomes was when I was a freshly minted PhD and I was starting my postdoc at Weill Cornell Medicine in Chris Mason's lab. And his lab was doing a bunch of environmental microbiome work, particularly in transportation systems. So I had done my PhD work in bioinformatics. The way of knowing that I had honed during that time of understanding biological organisms was through their DNA sequences. And I spent a lot of time thinking about plants. And then I joined the Mason lab, and everybody was thinking about microbes. And I was like, wow, we can use these tools of DNA sequencing to understand this invisible living component of our environment. And it was just fascinating to me because it's so counterintuitive. The built environment, or our city environment that feels very much built for human beings and occupied by human beings, also hosts a really diverse and vibrant ecosystem, but that we just can't see. But with this amazing technology of DNA sequencing, we can access that information.
Wow.
So just like our bodies and the different biomes on the planet have their own microbiome, so does this built environment. Does it just happen naturally? Is it because humans occupy these spaces that microbiome develops? Where does it come from?
That's a really good question. And the answer is going to be different depending on what aspect of the built environment you're looking at. So if you're looking at indoor spaces, most of the microbiome of indoor spaces corresponds to things that we as human inhabitants have brought into those indoor spaces. So the microbes that you're going to find indoors are mostly human microbiome related. A small portion, if you have, say, indoor plants, you might have plant or soil related microorganisms in those indoor spaces, but most of them are human related. Same thing for transportation systems. So the Mason Lab and actually the global consortium, the Metasub consortium, has been studying transportation systems for almost a decade now. So it's mostly human related. But other aspects of the built environment often reflect what that built environment was before it was geoengineered. If we look at a waterway, for example, we're going to find organisms that are related to a marine environment or related to soil environments for more terrestrial landscapes.
I'm sure pets and dogs influence it, because when I was talking to Ginger, she was telling me she had gone to a built environment workshop and they were talking about how having a dog contributes to a beneficial microbiome. Can you talk a little bit about what a beneficial microbiome is? What would that look like? And to piggyback off that, how do architectural and urban design choices influence microbiomes that flourish in cities? So both how are they beneficial and how do architects and urban designers even think about microbiomes?
The short answer to that second question is usually not, but I think more so now. A beneficial microbiome is perhaps tricky to be prescriptive about. So I couldn't give you a list of species and be like, you need species A, B, C for that to be beneficial. But what's kind of emerging out of a broad body of literature is that human health and well-being is really tightly related to our relationship with our microbiome. And what is generally beneficial for that is a diverse microbiome. So we can't necessarily determine specific species that would be best for everybody for the good reason that health looks different for different people. But in general, being exposed to a diverse microbiome is what's going to be beneficial. Those are usually hard to encounter in urban environments because urban environments apply pretty strong selection pressures against diverse microbiomes. And also the sources of microbiomes that we would usually find in, say, a rural environment just aren't present in urban environments. Microbiomes associated to soil systems or to plant ecosystems as well.
But can you game your urban environment or your home environment? And also game meaning, can you manipulate it in a way that it's going to be more beneficial, say, by either adding plants or adding a dog or a cat to it?
Yeah, I'll quote the great Jack Gilbert on this one, saying that the best thing that you can do for your indoor microbiome is to open your windows and have pets.
Nice.
Meaning really finding ways to create porous boundaries between the indoor systems and the outdoor systems. So pets being themselves their very own organisms with their very own microbiomes. They're going to be bringing in also the microbiomes that are associated with their bodies, but also because your dogs and cats are walking around outside and then coming inside and then you're petting them. And so there's kind of means of transmission for those diverse microbiomes. Same thing for opening your windows. So just being able to open your window and get fresh air. It seems too simple to be a technology or something.
Right.
But it is really effective. And for a lot of urban buildings, that is actually much more easily said than done because a lot of the buildings, either office buildings or high occupancy residential buildings, don't really have porous building envelopes where you can open the windows. In my office at NYU, I can't open the window. I can touch a temperature dial to adjust the temperature and the HVAC system will adapt, but I can't open the window. So interestingly, a lot of the progress that has been made in HVAC systems with respect to energy conservation, optimizing in that direction of energy conservation, so mostly recycling air within the building envelope, is actually detrimental if your goal would be to foster a diverse indoor microbiome.
Yeah, that is super interesting. I really want to dig into that. So let's put a pin in it because I also want to. You're like, there's so much to unpack. And earlier you mentioned the transportation system. So I want to just get that out of the way because we're going to go deep into the indoor built environment, which we spend a lot of time in. So you worked with Chris Mason and the Mason Lab on swabbing subways of NYC. Can you give our audience a highlight of what that was like and what was discovered in the subways and the microbiome of the subways?
Yeah, sure thing. Yeah, that was a really exciting project to be part of. So in that particular project, the goal was to swab every one of the 462, if I remember correctly, subway stations in New York City. So we were specifically swabbing elements of the subway stations, not the trains themselves, because the trains move around and so you lose that geospatial aspect of those samples. There are several interesting highlights to that paper. The main one being is that most of what we find in the subway from a microbial perspective is brought there by human beings. So microorganisms related to human skin, related to human guts, also related to the fermented foods that we eat. So that was one of the main takeaways, is that the microbiome of the subway system is a function of the input of human beings. One interesting takeaway with respect to design of the built environment is that there's a higher prevalence of antibiotic resistance in subway stations next to hospitals. So we know that antibiotic resistance, like multi-drug resistance, is a growing problem in hospital environments. So microorganisms who kind of develop the capacity to be resistant to multiple different antibiotic treatments. That's an example that could inform certain aspects of design of the built environment. So maybe we need to rethink entrances and exits to hospital buildings, apply negative pressure so that air isn't escaping, or use antimicrobial materials in the apertures and doorways of hospital buildings. So that was also an interesting takeaway. Another takeaway is that when you collect biomass from the environment with the goal of studying microbes, you also get DNA from pretty much any other kind of species that is in that environment. Those samples also yielded a bunch of human DNA related to the human beings who are taking the subway. And in sufficient quantity for us to be able to sequence it in a way to reconstruct reasonably accurately census information from the city. There was a good amount of human DNA collected in that process. It's interesting to be able to look at the multi-species assemblage, which is the subway, through this lens of DNA sequencing, but it also gives you a little bit of pause with respect to privacy concerns. Human beings are shedding DNA all the time in pretty much any kind of environment that we're at. And with the right kind of technologies, you can collect that DNA and draw conclusions from who has been where. I think we're not really talking at the moment of genetic privacy in public places. We talk about photographic privacy in public places, but I feel like this is a conversation that's going to come up very quickly.
It's been a while since I've gone back and looked at that study, but I seem to remember that there were some very unusual organisms that were found. And I wonder if you remember from a microbial point of view or a fungal point of view, what those might have been, number one. And number two, since you were collecting all this DNA, did you find any non-human organisms, rat DNA, dog DNA, cat DNA? Was any of that present? Crocodile DNA, the legendary crocodiles of the New York City subway system?
I'll answer that second question first. So the analyses that we do with these DNA sequences are targeted towards a particular hypothesis. So these are similarity-based analyses. So you have your kind of mixed bag of DNA sequences from unknown sources. And computationally, you take those unknown DNA sequences and then you compare them to sequences in known databases. And the computational approaches that you're going to use, you select ahead of time of which databases you're going to interrogate. There's a particular analysis that you run to interrogate microbial databases. There's another analysis that you're going to run to interrogate human sequence databases. And as far as I know, and we didn't publish this aspect of it, but I don't think this was even done as far as preliminary analyses. I don't think that we ran it against any other databases besides human and microbial. Surely we would be finding dogs and rats and avian, surely.
Pigeons, yeah.
I don't know if the pigeon genome is sequenced or not.
Yeah.
The funny thing, though, about that particular approach is that we don't have reference sequence, like genome sequences for all organisms ever. When you start taking these environmental DNA and comparing it to your database of known sequences, you'll often get hits that are not quite accurate. DNA that might be coming from, say, a plant that does not have a reference genome in the database is going to be reported as matching the closest thing that does exist in that database. So, for example, an agricultural plant, which are highly represented in our databases. So when you start asking really broad questions like that, you can actually get biased results because the databases that we have to compare to are themselves biased because they're incomplete.
Mm-hmm. Yeah, I mean, so to follow up on that, for the information or the DNA that was gathered, is it continuously being interrogated against databases that are being updated? Is there someone still continuing to work on this?
Oh, that's a good question. There's more publications that have come out of the Metasub consortium looking at this from a global perspective. One of the follow-up publications to this study was looking at the prevalence of antimicrobial resistance genes across the globe. So drawing antimicrobial resistance profiles of different cities across the globe. In that sense, you can reanalyze that data. When you have new algorithms or new hypotheses, you can go back to that data and reanalyze that with that in mind. To go back to that other question that you had with respect to interesting species that were discovered, There was another result from that paper that was really interesting and that sparked a research direction that I've been following in my lab, is that there was one subway station, the South Street subway station, that had been flooded during Hurricane Sandy, which had occurred a few years before that study, but not too long before. That study was in 2014. And so the subway station that had been flooded maintained this trace of marine related microorganisms. So even though the floodwaters had long ago receded and the subway was back in action and active again, there was this microbial echo of the history that happened in that particular environment. So that's something that really captured my imagination at the time and has stayed with me, of this idea that microbial species adapt very quickly to their environment, and the signal of that adaptation can persist over time. And so in a certain sense, being able to sample these microbial populations and see who they are, but also what they're doing, what kind of functions they encode, oftentimes gives you a read into the history of that particular site. Sometimes it's the history of human intervention. If you see these microbes have adapted to, say, anthropogenic contaminants or something like that, or it could be a history related to climate events like flooding.
This is so super interesting talking about that study, but you've been studying environmental microbiomes. And do you feel that knowledge has shifted our understanding of biodiversity in cities? You just mentioned a subway station that flooded that has remnants of the ocean microbiome in it. How does that change our understanding of cities? And do you think cities or global structures actually get it right when it comes to microbial diversity?
I think usually not, because we definitely don't have microbes in mind when we're designing the built environment. I think that if non-human or more than human living organisms are taken into account, we're thinking of macroscopic organisms, so usually thinking about green spaces in urban environments. Microbes are rarely taken into account except for pathogen surveillance and eradication. So the idea of designing the built environment with probiotic concerns in mind, I think has not quite latched onto the public imagination.
Yeah.
Though I think that we're certainly making progress in that direction. One of the hurdles is the fact that these are organisms that we can't see, so we can't really build a relationship with them. And measuring them is actually pretty difficult. Being able to get accurate metrics of a microbiome in the environment is quite difficult. That's one of the necessary conditions to be able to build that into a design pipeline, is to be able to get accurate metrics. To draw an analogy, we design spaces with a bunch of different environmental metrics in mind. We design for optimal temperature for human comfort, and CO2 concentrations in indoor environments, and light quality as well. These are all environmental factors that we have easy metrics for. We're able to have sensors that take continuous readings. We can model things like the thermodynamics or airflow in an environment. So those are all factors that we can measure and model accurately. And so therefore we can design for them. And microbes, we can't measure accurately. It's actually quite difficult. It's hard to model for them. So therefore it's hard to design for them.
Yeah. Yeah. To us who are entrepreneurs, it seems like there's a lot of opportunities to make for microbiomes and microbes, right? It sounds like on the analysis side, the measurement side, like you're mentioning, and then how we build our environments in a way where beneficial microbes and microbiomes are introduced.
Mm-hmm.
And we've been teasing it in the podcast, but I know this is an exploration that we've been going down, talking about what microbiomes exist in the built environment, but many built environments have been sterilized. I talked about hospitals earlier, and we quote unquote clean them, meaning that we just kill a lot of microbes with cleaning solution. So what do you think or know through your studies are the implications of such sterile environments on human health and the microbiome that we naturally interact with versus the microbiomes we naturally interact with in being outdoors?
Yeah, we certainly take a scorched earth approach to microbiomes in our
Yeah.
Why?
I think there's a lot of interesting cultural factors to that. And I trace it to the post-war cocooning effect. Post-World War II, there was a cultural shift towards isolation of the home environment with respect to the public environment. which came along with promoting nuclear family and this isolation of home environment versus community, and the idea of the home being the castle. There's a lot of interesting cultural artifacts that you can find also in the world of advertising with respect to cleaning products, which results from a lot of chemical industry development during the war with respect to producing chemicals for warfare. And once that was over, the industry had to kind of repurpose these chemicals that they were able to mass produce. So a lot of those turned industrial chemicals into cleaning products. You can see this boom in the advertisement for cleaning and disinfection products after the war. So there's cultural aspects of that. A lot of advertisements of white, clean, sparkling kitchens with a thin white woman presiding over it.
Yes.
So there's a lot to unpack there about why sterile and clean, like cleanliness, is associated with positive qualities. That's probably a whole other philosophical discussion for a whole nother hour. But there's a lot of literature that shows that lack of exposure to diverse microbial populations leads to negative health outcomes, particularly increasing the prevalence of non-communicable diseases, autoimmune disorders, for example, being one of them. And there's a lot of common practices that lead to that. So most of the studies had been correlation based, so not necessarily establishing causation, but there's a few really elegant papers that have been published within the last, I'd say, 5 or 10 years that show positive causal effects of interventions, so biodiversity interventions actually having positive health outcomes. One example of that was published, I believe the study was performed in Northern Europe. So the biodiversity intervention that they did was changing the ground substrate of a playground from that weird squishy asphalt stuff to wood chips. And wood chips being a bioreceptive material, there was a measurable difference in the diversity of the microbiome in that playground, and then measurable differences in positive health outcomes for the cohort of children who were playing in that playground. It's really amazing.
That's
Wow.
wild.
Yeah.
Yeah.
Hi, a quick shout out to our sponsor, Messaging Lab, the force multiplier for biotech. Your biotech company is making the world a better place. You know that, we know that. But does the world? There's a big reason why some biotech companies attract investors, sign up customers, and get attention. That reason is strategic communications. At Messaging Lab, we translate complex science and economics into compelling narratives. And we have done that for the most successful biotech companies across healthcare, agriculture, personal care and beauty, materials, and the list goes on. We're here to make sure your ideas not only get heard, but resonate with your audience. So if it's time to amplify your company's voice, visit messaginglab.com to explore how we can elevate your story and grow your business.
So tell us about your lab. You do projects that are this intersection of scientific inquiry and design. So it's a very interdisciplinary, multidisciplinary approach to help explore and solve problems around microbiome and human spaces. Can you talk about that? What are you guys working on?
Yeah, sure. We're working on a bunch of cool stuff. In general, we have a portfolio of projects that really use New York City as a living laboratory to start to understand that relationship between abiotic aspects of the built environment, so like non-living factors of the built environment, and then the microbiomes that inhabit it. So, for example, we have a project, we've been studying the microbiome of the Gowanus Canal for a long time now. That idea started in the Mason lab. That has just been one of my all-time favorite projects. So we're looking at the Gowanus Canal, which is a Superfund site, an EPA Superfund site, highly contaminated with anthropogenic contamination. So the microbiome that inhabits that contaminated sediment has adapted to that particular cocktail of contaminants and is able to degrade some of those toxic compounds. It's really interesting from a biotech perspective because that's a complex community of microbes that is able to degrade this cocktail of contaminants, and it is something that we would never be able to engineer in the lab. So we can engineer like a microbe to do a thing, but engineering like a community of microorganisms that lives and play nice all with each other and that also collectively encode these complex biodegradation functions is something that we wouldn't be able to make from scratch. So it's interesting from a biotech perspective, but also with respect to this microbial memory perspective. So we can look at the list of metabolic pathways that correspond to degradation of different contaminants. And then you can look at the historical data of the factories that have been in operation around the site. And so you can see a correlation between all of these, but also you can see microbial metabolisms corresponding to contaminants that are not present in the human records. The microbial records are in a certain way more accurate than human kept records, which kind of makes sense because illegal dumping isn't going to be reported and history is always written by the victor and human kept records are notoriously flawed. This idea of microbial memory to me is really interesting, and how it can be inscribed within their living DNA sequences, living genomes.
Wow.
We have another project actually that started with that sandy subway idea, with this idea of climate-related events being able to alter the microbial composition of a built environment. So with a colleague at Tandon, Andrea Silverman, we've been looking at the impact of street level flooding on microbial populations. So floodwaters in New York, we have combined sewage systems. So floodwaters are full of a bunch of different contaminants, biological contaminants from untreated sewage, but also chemical contaminants that are mobilized from the floodwaters. And so we have some interesting data, preliminary data that shows that the microbiome of the sidewalk will change drastically after a flood event. And then it takes about a couple of weeks for it to move back, to get back to its original configuration. So
Wow.
yeah, we're interested in anthropogenic change, contamination, climate change, also thinking about designing for indoor microbiomes. So I have a rock star PhD student who's no longer a PhD student. She's now a doctor, Dr. Phoebe Mankiewicz-Ledens, who led this really awesome study looking at how you can tweak design criteria for green wall infrastructure in indoor environments so that green wall infrastructure can seed indoor environments with beneficial kind of diverse microbiomes. So that's the array of projects from a technical standpoint. But I think one thing also that we're really interested in the lab is that for any of these ideas that we're developing within the scientific community to ever be translated to the public, we need to overcome this public hesitancy towards microbes in the environment. Most of the time when you talk about microbes in the environment, the reaction is like, yuck. For all sorts of good reasons, because they make us sick. Also, coming out of a global pandemic, we're all now very painfully aware of the pathogenic potential of invisible biological entities in our airstreams, in our environment. There's a lot of work to do to convey a public understanding of the science to be able to accept the idea of probiotic environments, of designing for microbes in our environment. And science isn't the best way to do that. Reading scientific articles is actually pretty dry if you're not really motivated to do that. So alongside the science, what I've been doing with members of my lab and then also industry professionals in artistic fields is producing artworks to try to bring this idea to the public about what does it mean to imagine microbes in an environment. How could we imagine building at a human scale for the microscopic scale of microbes? How do we push back against these power hierarchies between large multicellular organisms and the microscopic? That's another type of output that we really focus on at the lab is building art installations, and it's really fun.
Yeah, that's incredible. I am really curious. So you mentioned about the Gowanus Canal, that there's microbial fingerprints of what actually was dumped in the Gowanus Canal. What did you discover? And is there anything surprising? Were there bones, which I'm sure there probably were bones or human remains, because this is Brooklyn. Hello. But what type of contaminants? Are they PFAS? Is it effluent from certain industries that you're like, okay, and it was these microbial consortia that came together to degrade them. What contaminants and what microbes come together? Just to give us an example of what you were able to unravel from this investigation.
Yeah, sure. So the main source of contamination in the Gowanus Canal is related to energy production, specifically coal tar. So the sediment at the bottom of the canal is black and viscous and smells like gasoline. People call it black mayonnaise.
Ew.
It's heavily contaminated with hydrocarbons. And that's really the main source of contamination. There's also a bunch of heavy metals, and there's also PFAS, and there's also solvents. There was a lot of tanneries and paint making industry, but most of the contaminants are related to energy production. Those are the main metabolisms that we see.
And just so our audience knows, the Gowanus Canal cuts through northern Brooklyn. It goes straight to the ocean. It is a Superfund site. It's been investigated and supposedly being cleaned for at least a decade. There's also an enormous amount of construction going on around the Gowanus Canal. Something like 8,000 units are being built because it is right in the middle of everything. So it's super interesting to talk about remediating the canal, which has been something that supposedly has been worked on. But even despite the fact that it is not remediated, all this construction has been happening around it and really urbanizing the canal.
Yeah, it's a tricky situation. And it's a classic story of gentrification also, because I think it was declared a Superfund in maybe 2010 or 2013. And immediately after, property values skyrocketed because once it was declared a Superfund, then there is a plan and also funding for remediation and cleanup, which is currently underway. The dredging is happening now. The idea is that they're dredging as much sediment as possible and then sealing it with an impermeable layer and then putting the water back. I mean, the water is there and then calling it good.
Sounds a lame to me. I mean, I hate that, just cement something over.
Yeah.
Did they reach out to you in your lab since you guys have done so much research? I mean, hello, there's
No.
something to be done. I mean, I hear Superfund. I'm like, is there a lot of money going into this? Does it go into the pockets of people?
There is a lot of money going into it. And one interesting thing about the Superfund program is that the way that they fund these remediations is by holding accountable companies who have acquired the liability of the contamination. So usually that's the present day company who owns the land where the factories that were originally source of that contamination. And so the present day company is Con Ed. So Con Ed has acquired both the assets and the liability concern related to that contamination. But the question of remediation is really tricky. I think it's really hard to have black and white ideas about it because it's very much a question of time frame. So the microbes are doing their thing and they're remediating the canal, but given the kinetics of these reactions and the accessibility or lack of accessibility of those contaminants, it could take hundreds or thousands of years for that to go away. Maybe never. That's why tar pits exist naturally. And even though there's microbes that are surely consuming the hydrocarbons in those tar pits, they're still there eons later. That time frame is hard to reconcile with our impatient human time frame of state developers who want to sell waterfront properties, but also the real danger of exposure to families who have been living in that area for the last few decades where no remediation has been performed. The unfortunate thing is that those families are being displaced by the process of gentrification, usually historically underserved minorities. And so the families who have been suffering from exposure from that canal are not the ones who are benefiting from the remediation efforts.
You have these polluted areas throughout urban environments. In New York City, the other very polluted place is Newtown Creek, which is the boundary between Brooklyn and Queens. In your work in studying these things, how does it help people understand what's going on a biological level? Maybe not even the remediation, but just so that people understand. I mean, you mentioned that there's these consortia of microbes that are playing nice with each other and are also able to degrade these terrible chemicals that have been dumped there. But what do you do, or what can we do to help people understand what's going on biologically?
Yeah, I think that really is the role of artists and designers, is to be able to make that legible to the public, because I can make all of the data visualizations and graphs and plots that I want, but that's not necessarily what's going to tell a compelling story. And compelling stories is really what we need now. And I keep coming back to this idea that one of the hurdles is the fact that these are organisms that we can't see. And so they're just hard to relate to because we can't build an immediate relationship with them. And if anything, the relationship that we have is to the tool or interface that we have to relate with them.
Mm-hmm.
And just to give you an example of why I think design of collaborative tools or tools that aim towards collaboration with microbes are important, is that the most common tool that we have to interact with microbes is the swab. That's a tool that allows us to collect biomass from a particular environment, and then you take it to the lab and understand something about that. We're all very uncomfortably familiar with the swab now. And the thing is that the swab it's a clinical tool that was designed for a clinical environment, and it doesn't give us good vibes about what we're collecting with that swab tool. An example of that is there was a lot of people who challenged our undergrads while they were running around the subway collecting these swab samples. And a lot of those reactions were to the tool of the swab. People were like, oh gosh, is there an epidemic? You're monitoring for an epidemic. This was pre-pandemic. There was even a team that was accused of implanting HIV in the subway. And that reaction was very much to the tool because it looks scary. It belongs in a clinical setting. It makes us think of contamination and illness. But at the same time, you could take a microbial sample by just grabbing a handful of dirt. And if you think about that gesture feels very different from the gesture of taking a swab. Swabs make us think of illness, and then a handful of dirt makes us think of groundedness and connectedness with nature and has all sorts of positive associations. I think really the role of communicating, the role of artists, the role of designers is to be able to think about creating interfaces or means of interaction with the microbial world that carry this positive valence and connotation, as opposed to the tools that are most frequently at our disposal, which carry this negative connotation with respect to that interaction.
Yeah, I mean, I love that. Wow, that is so powerful. What are the ways to make the invisible visible? What a great design and exercise. And there's so many tools now on the scientific side, but how do you make them more user-friendly, more understandable? So we talked about microbiomes in the water and soil and subways. You and I and Ginger and Karl and I had also talked about the microbiomes in the air. And you guys have been doing an excellent study of the air biome, I think in Brooklyn particularly. And you guys have come up with some very innovative ways to examine the air biome, not using swabs, but using either another object or just looking at nature in a very unique way. Can you share a little bit about your air biome project and how you guys went about it? And then one other thing I would love for you to talk about, I'm just going to say 2 words: red honey. Go.
Sure. It's all connected. Yeah, aerobiomes to me are just fascinating. When I realized that you can think of air as a fluid medium that is also carrying biological organisms, it just totally blew my mind. I was like, oh my gosh, they're everywhere. What we think of the void is not void, and it's actually full of living organisms. Awesome. Sampling the aerobiome is particularly tricky because it's not very dense. So there's the similar challenges sampling air as we have sampling water, actually, that you need to sample large volumes of it and concentrate it into a small volume to get some kind of insight from it. So in the lab, we've developed a couple of different approaches to that. And with the perspective of having the approach adapted to the scale of the thing that we want to study. So coming back to our tools being our means of interaction and the swab being the tool that we use most frequently, a swab can sample an area of maybe 20 by 20 centimeters. So we're talking about square centimeters in scale. But if we want to study, say, the microbiome of an entire neighborhood or even a city block, then that tool, the swab, its resolution is not adapted to the size of the thing that we want to study. We've invented a couple of tools to address this question of scale in the lab, starting with large scale. This was a collaboration with Kevin Slavin when he was at the Media Lab, and also started in the Mason Lab, where we were like, what can we do to sample the aerobiome of an entire city block or of an entire neighborhood? And went through a bunch of ideas. And this was the time when there was that news article in the New York Times about the red honey. So these honeybees that started making red honey, and the beekeeper couldn't figure out why that was the case. And it turned out that they were going to forage in maraschino cherry factory. And then there was a health inspection about why are there honeybees that are getting into this cherry factory in the first place. And then it turned into a whole nother thing where actually the cherry factory was a front for a large weed growing operation. And anyways, it was a big story.
Yes. Yes. I just love the fact that biology and science, and you're investigating something, is uncovering all of these crimes. True Crime is the top podcast.
Yeah.
True Crime Biotech podcast. Like, that's next year. We got to do that. Well, that
That's
with
a
you.
good idea. Yeah.
But yeah, I
But
just love
what came
that.
out of that whole series of news stories is that bees are very much reporting on what they interact with in the environment, and their foraging range defines what they're able to report back to. So we observed that these honeybees were basically doing what we had been asking the hordes of undergraduates to do for the subway study, which was go out into the world, interact with things in the city, and then always come back to the same place, which is the hive or the lab for the undergrads. That sparked this wild idea where we were like, gosh, bees are really, they're out foraging. They forage in about a 2-mile radius. This is very much an average, depends on nutrient availability and things like that. But it's quite large radius at the scale of New York City. And they always come back to this very specific place. Our hypothesis was that anything about honeybees, what they produce, honey, propolis, etc., or anything about their hives that would give us a readout of the microbial environments that they're interacting with. So we did a whole pilot study, and the conclusion of that pilot study is that the debris that accumulates at the bottom of a beehive can be queried for microbial signatures of the microbial environments that bees are interacting with as they go and forage around. And so we figured that we would find plant microbes because like bees and plants and flowers, that all makes sense. But it turns out also that we were finding microbes related to all sorts of other aspects of the environments that they were traversing. So bodies of water, if there happened to be aquatic microbes, if there happened to be some kind of body of water in their foraging range, microbes related to humans, microbes related to our pets. So that totally reframed our idea of what was happening in the bees' environment, is that this idea that even though they weren't necessarily physically contacting these different things, like going into the pond in Prospect Park or landing on a dog or something like that, they were traversing these microbial clouds, to use a Jack Gilbert term again, as they were going on their foraging trips, and then bringing all of that information back to the beehive. That's a study that then figured out a protocol about how to figure out these microbial signatures from a beehive, and then did that globally. So we had collaborated with beekeepers here in New York, in Europe, Tokyo, and Australia, and published that paper just a couple of years ago.
So you're saying that if we have beehives every 2 miles in a place like New York City, we could really have a much better understanding of what's going on microbially than having a bunch of grad students running around swabbing every surface.
Yes. Again, it's tuned to the scale and resolution of where bees are at, but that tiling would work quite well. And so Tokyo was the city where we had the most dense layout of hives. And so we could pretty much correlate geographic distance with microbial distance. So the further those hives were away from each other, the more different those signatures were.
It's amazing.
Wow.
That was
Yeah.
a really fun project. Obviously, that works only during bee season, which in our part of the world is May to September.
Wow.
Yeah.
So that's a published paper, so we can link that in the show notes. People can explore more. Yeah, that's just incredible. I just love the level of thinking and just how many leaps of thought had to happen to be able to just have the hypothesis of looking at bees. It's just incredible.
I mean, that's what happens when you work with an interdisciplinary team. And a lot of those early conversations were with artists more so than scientists.
Wow.
And Kevin and Chris and I wrote up a little behind the paper article explaining the genesis of this project. This project and its data were shown at the Venice Biennale before they were published.
Amazing. Amazing. We've talked a lot about the microbiome of the built environment, and we're seeing this trend towards the building of our ecologies. I think of the line in Saudi Arabia. These generally would be, like you say, a very potentially sterile environment. But how do you think an environment like that, or as we start to like even maybe climate will force us to create these bigger arcologies around New York City or in Nevada or wherever, how should designers of these places be thinking about that microbiome that's going to be living in there?
Yeah, that's a tricky one because the insight that I have from the Gowanus Canal is that the microbiome that we find in that environment, we couldn't engineer in a lab. I think it's also the case that when we think of a beneficial microbiome for human health, we can probably also not engineer that in the lab. So I don't think we're going to have a room spray or something that would be effective to maintaining a diverse and stable microbial ecosystem in an indoor environment. I think we're going to have to think holistically about fostering diverse ecosystems in general across all scales and trophic levels, and that in that process, we're also fostering a diverse microbial environment.
And it sounds actually like it can be pretty simple. It's open your windows, have a pet, and have some plants around.
Yeah. And have diverse ecosystems outside of your window, too.
Yeah.
Yeah. And then go out into the world more.
Go out into the world. Go hang out in the forest.
Yeah, go hang out in the forest. Listeners might be going for a run. That'd be great. But if they're listening indoors, get outside. Go out there, lick a tree. I don't know what would happen.
But hug a tree.
Yeah, hug a tree, hang out.
Not health advice. Not health advice.
Oh, yeah. Please consult your physician.
Yeah, before you go licking Subway.
No, I don't advise that. But there's a reason why we feel good outdoors. Biophilia is a real thing. There's a reason why it feels really good to go for a walk in the forest. And I think that surely part of that feeling good comes from our body registering the fact that we are being exposed to these diverse microbial populations. We've talked a lot about tools and how we have this push towards developing technologies and modern objects to interact with these microbes, but we've also never not lived in a microbial world. Our understanding of microbiology as being a recent thing that has been developed by Western biotechnology. We've known about DNA for 75 years now, which is a very short period of time. And that's one of the main bases of our understanding of microbes. So it feels like this is a fresh new thing that we've discovered only since we've had these biotechnological tools, but we've never not lived in a microbial world. And I do believe that there are ways of paying attention to our environment that surely have been lost in the society, at least that I live in here in Western society. But traditional ecological knowledge certainly has ways of interacting and mediating this microbial relationship that long predates our Western concept of biotechnology and the tools that we've developed. Coming back to that idea of why does it feel good to be in the forest, I think that there's means of close observation that we could cultivate that are also ways of interacting with the invisible living component of our environment that don't necessarily need the high-tech approaches of DNA sequencing.
Totally.
Oh my gosh, that's a whole nother podcast you just started. Just the indigenous approaches to living healthy and their understanding of microbial world, or this invisible force, I guess you can say. That was at the time that we are trying to make more visible with biotechnology, but it's almost like we're starting from scratch because we've lost a lot of that wisdom. But we're getting there slowly but surely, hopefully. But thank you so much, Elizabeth, for coming on the podcast, sharing your work, the work that you're doing at your lab, the work your students are doing and your collaborators. It's so very exciting. I know you're going to be up to more and more exciting work in biology, art, design. We'll have to bring you back on because we just scratched the surface, whether it's sterile or it has microbes on it. We just scratched it, but we're going to go deep. But I'll see you soon again, I'm sure, just being around the world of Brooklyn. And again, thank you so much for coming on.
Thanks,
Sure
Elizabeth.
thing. My pleasure. Great talking with both of you.
Thank you.
So, Karl, what'd you think of that episode?
Elizabeth is awesome, and I'm really looking forward to hanging out with her in real life, in person, at one of our future events. There were so many things she said that were so profound. Just thinking about the built environment or the environment that we live in, you could see that some of the questions I asked were really about, hey, we live in these concrete boxes. What are we bringing into them? How does our microbiome affect them? I had all those questions about dogs and cats and how those make us more healthy. So I just loved it. What did you think?
Yeah, this was the moment that I've been waiting for. I've been wanting to get her on the pod for a while because we've had such interesting conversations, and I'm happy that we were able to bring that to our listeners. But using NYC as a living laboratory, really looking at the different environments which we live and breathe. So from the subways to near the Gowanus Canal. And she's done all this work with other amazing scientists in New York City. So the subway swab was a part of Chris Mason's lab, and she was a PI there. And we'll have to have Chris Mason on to speak more about it. But it was cool that she shared some of their lab's work.
I think about it when I see people eating on the subway. I'm always like, that is so gross. I would never want to eat anything. I wouldn't even want to drink water. We all do it. I never eat in the subway. It's disgusting. But then a lot of times you'll see people putting on makeup when they're in the subway. But whatever's in the subway is floating around and you're putting that on your skin or near your eyeballs. So that's gross. I'm sorry.
Yeah, I still wear a face mask. I'm one of those people.
I need to do that.
Yeah, I still do. Mainly when it's rush hour because there's just a lot more people there. It's just hard to predict. And it's not that big of a deal just to wear
it's not
during that time. But I want to learn more about it. So we'll have Chris Mason on to talk about it. But Elizabeth was a part of that. So that was a part of her inquiry into NYC being a living laboratory. She also mentions they were able to also find human DNA and were able to reconstruct the people using their DNA, which then brought up privacy concerns, because if your DNA is like anywhere and whoever swabs the DNA can reconstruct that, that's Black Mirror territory. But it's also that, okay, that's our DNA, but we already live in a surveillance state. There's cameras everywhere. I didn't consent to a camera being in Times Square, which I rarely go to, but it sees that I walked around. It's a great ethical thought exercise on what is right and what's wrong. But it is. I mean, maybe people are like, I want to live my life and I don't want people to know like my whereabouts. So I thought that was interesting.
Yeah, well, people will know your whereabouts if you're carrying your phone. So there's no getting around it. But it brings up two things. One is not consenting to having your DNA swiped. I think we should link to it. I know we've mentioned it in the past. There was an Atlantic article that Andrew Hessel co-authored back in 2011, I think, called Hacking the President's DNA, which talks about having a swab of the president's DNA and how you can make the president very sick if you were able to hack that. And it's a pretty elaborate story. The other thing is like this ability to being able to reconstruct someone from those random DNA swabs. There has been art projects where people create faces based on this DNA. I can't remember who the artist is. I think about it a lot. Like when people hand me a business card, I'm always like, oh, I wonder if I could reconstruct this person based on, what I swab off this card. And given that one of the only things that I hoard are business cards, I've got decades worth of business cards. How many people could I reconstruct off of those? But a lot of stuff to think about. And I'm glad Elizabeth prompted that in our conversation and now as well. And I'm sure she'll do the same thing for the listeners.
Yeah. I wonder if she worked on that, the one that you're thinking of, the artistic exercise, because I think that was in the Smithsonian. I just wonder if it's related because it's reconstructing faces based on random DNA is just, I don't know how many people are doing it. So I'm just curious.
I don't think there's that many people doing it. This is what I love about being in biotech and having people say we need the culture. Like Alex Titus said it a couple of weeks ago, the culture helps us understand these technologies. And I'm a big believer in going to art galleries. Of course, my wife is an artist, so that's part of the reason why. But we need that creativity to get us thinking about things. And when biology is a part of it, then it can help us understand how these technologies are going to be used or will be used. And I think that's super important for us.
Yeah. So this was more of a art exercise or a scientific inquiry, but she also talked about the microbial memory. DNA swabbing, microbial memory, discovering crime scenes. This is my dream come true. This is like forensics, right? And I was just like, wow. And you're talking about culture. The biggest cultural phenomenon in our podcasting world is true crime. It's true crime.
Right.
So, I mean, we talked about having a true crime biotech podcast. If it's not us, hopefully one of our listeners start that, and we're happy to be guests on that or participate in some way. But so much we can learn just by looking at microbes. Come on, people. We need all these technologies to be built. I mean, I met someone that was working on creating very rapid biosensors to detect different microbes, from bacteria to fungi to anything, just handheld. And I was like, yes, we need some of that. Just very fast things. There's devices like the Oxford Nanopore. We can sequence eDNA or different DNA in the field. We need more of that so we can just illuminate what's going on in our world. I'm just very excited for what's to come, and that we will talk about it as soon as we find out. But if you, our listener, know about very innovative technologies to help us really understand what's going on in the microbial world, let us know. We really appreciate it.
So this podcast drops the Friday before Thanksgiving. We're going to be doing a replay episode, and we'll reveal that next week. We are super happy always to get feedback from our listeners.
So I wanted to highlight a comment that we received for the podcast from one of our listeners, Stephanie Azopardi. What Stephanie said about the Grow Everything podcast is, this podcast has opened my eyes to the possibilities of what biotech can accomplish beyond the biopharma world. Karl and Erum artfully interview thought leaders who are at the forefront of bringing new sustainable solutions in the face of climate crisis. Very engaging conversations and inspiring stories that open up the world of Symbio to noobs or newbies. So thank you, Stephanie, for leaving a comment. Please, listener, share your comments, share your thoughts. It's always helpful. Please rate, review, and subscribe. That is the Grow Everything podcast.
Thank you, and we will talk to you soon.